We are all familiar with CELMoDs. The idea here is to review a little bit these compounds, the current compounds in development, some data regarding their activity, and discuss how we think this family of drugs currently fit into the landscape and what are the purpose. So I think, the CELMoDs have had a previous name, but basically these are drugs that are binding to cereblon, and cereblon is a substrate receptor for an ubiquitin ligase, a choline-4 ring A3 ligase, and they target protein for proteolysis through the ubiquitin proteasome pathway. I think you are all familiar with that. And what these drugs do is that they modify the conformation of cereblon within the A3 ligase, and depending on how this configuration is and conformation is modified, eventually there are new substrates that are taken and brought to degradation, including sometimes some lymphoid transcription factors such as Ikaros and Aiolos, which are important for B cell survival, immune evasion, and have also an effect on T cell function. So it's really like an immune modulator drug or immunotherapy, which is original by itself. And I think you have the classical scheme of how it works. So they have, this new generation, I will say, of drug, which are called CELMoDs, are distinct and improved from the IMiDs that we knew from years. The molecules are larger, they have a higher affinity binding to cereblon, more interaction sites, and most of them have, a stronger immune responses and greater effect on T cell activation, NK cells, and also tumor cytotoxicity. This is obviously a summary of a lot of preclinical data in different models that is shown here, but basically, again, this is the efficacy in terms of substrate degradation. So I think the three drugs we will briefly discuss, and mostly the third one, are drugs you have been involved in some of the development after, obviously Revlimid and pomalidomide in myeloma. But this is avadamide, iberdomide, and golcadomide. And you have here, the structure of these drugs, the modification of the structure based on these interactions. And you can see that there have been further and further modification. There are, for some of them, demonstrated efficacy in multiple myeloma, particularly iberdomide, and are assessed right now as single agent or in combination. So I'm not sure everybody is familiar with this iberdomide data. I think, Catherine, you presented them actually 2 years ago at the ASH meeting. And this was a phase 1 trial with assessing safety, PK, efficacy of iberdomide as single agent or in combination with either rituximab or obinutuzumab in patients with different subtypes of relapsed refractory lymphomas. And these were patients treated with multiple agents, heavily pretreated, including some patients having received CAR T cell therapy. So you can see the graph bar with the response rate in the different things. We don't have the breakout by histology, but as you can see, the response rate range from 38 to up to 63%. A higher response rate in combination with rituximab, something we are familiar here, single agent, again, 38%, with 29% of CR rate, and again, an increased overall response rate, and probably the same CR rate when we look at the combo with different drugs. The toxicity is the one we know and have seen with these drugs. A couple of hematological toxicity, including grade 3-4 neutropenia, some anemia and thrombocytopenia, very few febrile neutropenia, and some others such as GI effect, asthenia, pyrexia, cough, and back pain. I think probably you are more familiar with the golcadomide data, which came from multiple phase 1 and phase 2 study, and which has an active development. There was a large study, which was multi-center, first in human open trial, which assessed the safety and efficacy of golcadomide alone or in combination. So this is again, the classical inclusion criteria for third-line study, I will say. There was a dose escalation part, there was a golcadomide monotherapy, and there was some dose expansion, either as single agent or in combination. So these are the efficacy data. They were presented in different meeting in 2022. They may have been updated. And this is the last data cutoff that is available. Probably the reference needs to be updated here. But if you look in follicular lymphoma, this is as a single agent an overall response rate of 67%, including 25% of CR rate. So this is an interesting agent as single agent. It's probably over what we might observe with lenalidomide alone. If you combine it to rituximab, you see that the overall response rate is higher. And then we have, different doses that have been explored and different schedule with rituximab, as you can see. And, the overall response rate range from 72 to 100%, and the CR rate, from 24 to 37%. And there is a couple of patients that have been len exposed, and it seems like we have a good response for those, and a good response in patients that have been exposed to CAR-T. So overall, I would say an interesting response rate in the follicular lymphoma landscape as a single agent or in combination with rituximab. If you look at the DLBCL cohort, these are a number of patients treated in combination with rituximab. And you can see that there seemed to be a dose effect similar to what we have seen in follicular. When you go to 0.5 milligram plus rituximab, there is an increase in the CR rate and the response rate. So if you look at that in these 35 patients, you have a CR rate of 43% and 17% of partial response, which brings us to an overall response rate of 60%. If you look at patients treated post-CAR-T, this is a 50% response rate with 33% CR rate. And in the CAR-T naive, this is an increased response rate, and with half of the patient reaching a CR. Obviously, these are phase 2 data, small numbers, but I think there is a signal of activity in both follicular and DLBCL in quite heavily pretreated patients. In terms of safety, you have here the safety according to the different cohorts and the different dosing. And as you see, the majority of patients have some side effect. I think what is notable as a major side effect is obviously neutropenia, which is very frequent as a single agent. And what I found interesting is that in follicular, there is not much difference in the incidence of neutropenia according to the dosage of 0.2 versus 0.4 milligram, while clearly in the DLBCL cohort, the 0.4 milligram has increased toxicity. The febrile neutropenia incidence rate is rather low in follicular, substantially higher in DLBCL at a higher dose, but this might be also related to the patient population. There is some grade 3/4 anemia and thrombocytopenia, again, probably a little bit more frequent with a higher dose. And the side effects are the usual side effect of IMiDs. And this is an ongoing studies with some data still being accumulated in these cohorts. So some of you may have are already participating or may think to participate to this study, which based on the efficacy on DLBCL, there is a phase 3 trial that is being conducted with a Golca R-CHOP versus placebo R-CHOP. The golcadomide is given at the dose of 0.4 milligram, day 1 to 7 at each cycles of R-CHOP. And this is 6 cycles. The primary endpoint is the PFS by investigators. Your secondary endpoint are classical. These are patients with an IPI score over 3 or an IPI score 1 or 2 with LDH over 1.3 upper normal median or bulky disease. So a little bit kind of mixed bag of patients with either high IPI or medium IPI with other criteria. This is classical large B-cell lymphoma patient, 850 patients, and obviously a registration trial for this drug in the frontline setting. In follicular lymphomas, there is a parallel study being conducted, which is also untreated follicular lymphoma patients with golcadomide plus rituximab at different dose and different schedule here. This is 2 weeks out of 4, and this is compared to R-CHOP or R-BENDA. And again, it's a randomized trial with the primary endpoint being a complete metabolic response and an estimated enrollment of 90. So I guess this will be more like a phase 2 randomized study to gauge the efficacy of this combination as compared to the thing. I think the randomization is probably 1-1-1 because there are 3 arms in the study. So I think this is basically a summary of the ongoing studies. As we said, you know, the phase 1 of iberdomide is probably getting close to the end. There is an iberdomide or golcadomide plus R-CHOP phase 2 trial in ABC DLBCL. These are phase 1, phase 2 studies. There is a couple of combination being assessed with different drug. But I think with golcadomide, as you can see, this is for other active regimens. These are different trial with different combination. I mean, some of the combinations are a bit complex, I will say, or involve also other agents before, after CAR-T or immune checkpoint blockers. But I will say these are small studies compared to the 2 large randomized study we have seen.
Hi, good morning. Maybe I will start. You know, I think those drugs really synergize well with the antibodies based on mechanism of action. And that's basically a rationale for combinations which you had shown. And the activity of those drugs look actually much more promising or much more potent than what we had seen previously with IMiDs. So golcadomide really shows excellent activity in combination with rituximab. And in this combination study with R-CHOP, the frontline study, looking at the pilot study, really resulted in a higher level of MRD negativity than historically would expect in those high-risk patients. So I think, you know, based on the synergy of antibodies, it's really a promising backbone in aggressive lymphomas. That's mostly in combination with chemotherapy. In indolent lymphomas, it can provide this chemo-free backbone in combination with other antibodies, in possibly even a frontline setting over time.
Thank you, Greg. Any other insight, Catherine?
Yeah. Oh, sorry. Could I just ask a question? You know, the efficacy is very intriguing and, you know, looks exciting. But one of the questions has to do with the toxicity profile. Can you comment on thromboembolic phenomenon and any teratogenicity?
I personally don't have the detail of this data. Maybe Greg or Catherine, who have been more involved in the studies. I think there have been no thromboembolic event mentioned as a significant number of cases, but maybe a very few cases. And obviously, teratogenicity, I guess these drugs have the same level than the classical drugs that target cereblon. But maybe one of you may have the answer for that.
The same answer, and there is the obligation to be really cautious about teratogenicity. And regarding thromboembolisms incidents, there is no data, major data regarding these events. I have one question for Greg or for Gilles, or both of them. Do you think that we could expect to have difference in terms of response rate between large B-cell lymphoma subtypes ABC and GC when you combine golcadomide and R-CHOP? or not at all?
That's an excellent question, Catherine. I think with lenalidomide, we're expecting more activity in ABC subtype or non-GCB subtype of large cell lymphoma. Here, in particular in the frontline setting, in combination with chemotherapy, it appears that it's actually active across the subtypes. That's true about the relapsed refractory setting as well. And possibly it has to do with this mechanism of action, primarily working for ADCC and enhancing the antibody activity rather than direct cytotoxic activity, which is typically seen in ABC subtype. So I see them as agents kind of working across the molecular subtypes of large cell lymphoma based on this synergy with the antibodies.
Yeah, I agree.
I think we have comment from Marek and then from Uli.
Yeah, thank you. So I have let's say two questions. One is a combination with the bispecific antibodies, because we know that it seems it works well with the rituximab or obinutuzumab. This is the first question. And the second one is if you see any place, for instance, to substitute lenalidomide by the golcadomide, for instance, in the Viper regimen, because of the toxicity and the mechanism of effect. The Viper doesn't work very well in the GCB, so it's at least based on the published data. So any comment on that?
I think, again, I'm not here to answer all the questions. I'm not sure that Greg or Catherine, who have been more involved in the trials, do have these answers. I think there are ongoing phase 2 trials assessing the combo with bispecific in different settings. And I don't think data regarding this efficacy have been yet presented, and toxicities. And I think the question you raised regarding GCB is really interesting one.
I agree with Marek. I think with bispecific antibodies, this could be really a potent combination. So the studies are ongoing, and we'll quickly learn about combinations there. In terms of the Viper, that's, the toxicity profile is a little bit different. It does cause some neutropenias, I think, and then cytopenias in general. So I think you would almost need feasibility pilot study to actually show you can combine it with the drugs in Viper. It sounds like an IIT in Czech Republic to run.
Uli?
Yes. I have a comment regarding the lines of therapy. I mean, I think this is definitely an interesting agent beyond second line. I think there we probably have a lot of applications, including post-CAR-T or during CAR-T or whatever. For the first line, if you look at the design of the study, IPI3 as an entry criterion in my country will definitely treat these patients with R-polar chip. So the control arm is R-polar chip. And I think that's something to evaluate if we are really happy with putting it into first line.
That's an interesting comment. And before handing that to Miles, I will say that this is another study in the first line setting. And, we try to review a little bit the landscape in a paper recently with David Qualls and Philippe Barmont. But we may have to think that unless one of these studies really show, a major breakthrough in terms of CR rate and PFS improvement, OS improvement, we will have a couple of data here, and there will be other arguments to probably use one compound versus the other, including practicality, toxicities, and things like that. Because, we have a backbone, whether it's R-CHOP or polar chip, it's another debate, but I think let's imagine 3 studies runs to be positive with bispecific, with Golca, with maybe Tafa, Akala, and how are we going to choose? And I think that's an important. Miles, you raised this hand. You raised your hand.
Yeah, thank you. Yeah, look, I think they're fascinating molecules, and I've got a couple of comments and questions as well, because we use these molecules in myeloma, has been interesting to see how they've developed over the years. This is very much to use iberdomide early in disease in myeloma, and mezigdomide, another CELMoD, for late disease. And it's very much a strategy. It's more strategic than necessarily deciding, which is better in those two situations, because both of them are significantly cause hemopoietic toxicity. And I think the big challenge when using this up front is going to be, are you going to compromise the CHOP backbone? We found that with romidepsin in T-cell lymphoma, that we had to dose reduce, and that ended up probably, giving us a limited efficacy. And so I wonder what the comments are about, trial design and making sure that we're optimizing the CHOP backbone when it's used up front, because it seems to me that the trials, at least presented, are tending to using these molecules earlier.
I can maybe comment about the dose since I was involved in a phase 1b escalation study. So it's kind of interesting because the study was actually done with the feedback from FDA with the Project Optimus, and we sometimes complain about dose optimization, but really I believe it serves us well before launching those large phase 3 studies. So we really have a dose workout, and in this case, actually, there were several dose levels studied. And for example, the longer schedule of 10 days was actually resulting in some dose delays of R-CHOP, and this was abandoned. So this dose and schedule, which you see in phase 3 here, was basically chosen based on a totality of evidence, meaning the efficacy was improved when compared to historically how those patients would perform with this IPI, but also the dose delivery was great, and there was no significant delays of R-CHOP. This was the major criteria why this dose was selected. And I absolutely agree with you that, when we're launching those large studies, we really have to make sure that it's not jeopardizing the chemotherapy backbone. And in this randomized phase 1b study, this was done.
Francesc?
Yeah, hi everybody. I was thinking that this drug probably would be placed in follicular lymphoma, substituting or instead of lenalidomide. Do we have data supporting that this drug is better or less toxic than lenalidomide in follicular lymphoma? I'm thinking about the regulatory standpoint. So why should we use that drug instead of lenalidomide in follicular?
I think if you look at the data that I have presented briefly, I think as a single agent or in combination, at least single agent, I has a feeling that it's more efficient. And there were a few patients that were exposed to len and still responded. So I think it's likely to have significant activity, and probably whether it's a direct effect on B cell or whether it's an effect on the immune cells remain to be discussed. The question is, how do you fit in the landscape? And that brings very well the comments from others regarding, the drug development is very complex those days, and the approval path in the phase in the second and third line is complicated, and that led the companies to try to move that drug very early in first line to try to access the market. But I think in follicular, there's a good chance that it's equal, if not better. After that, the company knows very well the issues that happened with relevance, where, building a superiority study was thought at that time to be too consuming in terms of money and patients, and basically didn't do an equivalent study. And the superiority was the aim, and we, instead of doing an equivalence, which will have needed a few more patients. So what the strategies that will be adopted based on the phase 2 is interesting, but I think there is a role probably here. I think we have to wrap up in 1 or 2 minutes. Is there any comments, Sonali, Karen, that anybody wants to make? Marek.
Yeah, you have shown that the combination with rituximab, the iberdomide plus rituximab, is almost equal effective as a combination with obinutuzumab. Do we have any data with the golcadomide plus rituximab versus golcadomide plus obinutuzumab from early phase studies?
Not sure we have those.
I don't think so.
Thank you.
the interpretation of the rituximab lenalidomide versus rituximab obinutuzumab versus lenalidomide obinutuzumab has been complex. The Galen versus R square, and I think there is probably a general feeling that while both regimens are working and efficient, there is no very significant difference in the efficacy of these both combinations. So I think obviously Obin is more prone for ADCC. It's probably also a much stronger a depleting agent in peripheral blood and marrow, showing superiority in CLL and probably in mantle cell by indirect data comparison. But whether in follicular or large B cell, it changed something, as we know the result of the data in randomized study are not supporting that.
And if I can follow your comments, so I think that basically on the mechanism of action and the combination, it seems to be promising, or could be promising in the mantle cell lymphoma as well, maybe more than the R square. Any
good option to
trials?
explore. Again, I think part of the difficulties, personally, my feeling about this drug is that they show efficacy clearly. I mean, I'm talking about Golka essentially. They clearly shows efficacy. It's an interesting efficacy. We are now close to a threshold where, the neutropenia associated make it a little bit more complex to combine. And what exactly is the clinical endpoint you want to achieve to make a meaningful change in the landscape? That this drug works, has efficacy, there is no question. The question is, where does it fit in terms of combinations as a way to be approved, to be being part of a landscape which is highly competitive those days. Okay, I think unless there is other things, I mean, we heard a lot of things regarding the potential of this drug, the cautions about the side effect with limited data right now in phase 1-2 trials, the interest in the randomized trials, the interest in some subgroup of patients regarding ABC or mantle cell, and how it will be challenging, but at the same time interesting to compare the efficacy of this drug as the one obtained with lenalidomide, which is part of the standard landscape in follicular R-square, is a major control arm of all second-line trials. So probably this will be important to assess for the future based on this data. I think we should probably close here and move to the other discussion.